This is the lecture for Chapter 13, Viruses and Prions, Part 2. In this part of the lecture, I'm going to focus on the animal viruses, and then at the end, I will go over prions. So as a review for the structure for animal viruses, all animal viruses must have a capsid, which is composed of Pap smears, and they must also have some sort of nucleic acid. So the nucleic acid could be DNA or RNA. Also, some animal viruses may have an envelope, but that is not required for all animal viruses. So all animal viruses will have some type of capsid and some sort of nucleic acid, and some may have an envelope. Like bacteriophages, animal viruses also go through the 5 stages of the viral life cycle. And as you remember, those stages are attachment, penetration, biosynthesis, maturation, and release. And knowing the structure of the animal virus will allow you to determine the details of each of these stages. Knowing the outer covering, what is the outermost layer of the animal virus, will help you to determine how it attaches, penetrates, and releases from the host cell. So all of these are dependent on what is the outermost covering. Is it just the capsid, or is there an envelope? So knowing that about the virus will give you information on how it attaches, penetrates, and releases. Biosynthesis and maturation, this is determined by the type of genome that the virus has. So is it a DNA virus or an RNA virus? Knowing that information will give you information or give you the location of biosynthesis and maturation. So in determining the complete viral life cycle of an animal virus, you need to know what the outer covering is. Is it a capsid or an envelope? And you also need to know what type of genome does it have. As long as you know those 2 pieces of information, you can give a full viral life cycle. The first thing I want to do is go over how the outermost covering affects the life cycle of an animal virus. And the 2 types of outer coverings are either just having a capsid or an envelope. And if the virus has just a capsid, I refer to it as a naked virus. So that's also known as a non-enveloped virus. So a naked virus or non-enveloped virus is just a capsid on the outside. There is no envelope. So this outermost covering is made of Pap smears, which are proteins. And remember when I talked about the viruses before, on these, especially on polyhedral viruses, but on all naked viruses, you have these protein fibers that are sticking out from the corners. And this is what the virus is going to use for attachment. So that first stage in the viral life cycle for a naked virus, that virus will use these protein fibers to recognize and adhere to the host cell. The next stage in the viral life cycle is penetration. So for a naked virus or a non-enveloped virus, it will use receptor-mediated endocytosis as its method of penetration. And remember, penetration is how the virus gets its genome into the host cell. Receptor-mediated endocytosis means that the virus, and we'll pretend that these purple triangles are the virus will bind to a receptor. So the virus will use those protein fibers to bind to a receptor, and that will send a signal into the cell to perform endocytosis. So the cell will actually perform endocytosis, moving its cytoskeleton and membrane to pinch off a vesicle, and that brings the virus into the cell. So the virus is basically tricking the cell into taking it in by endocytosis. So again, this is receptor-mediated endocytosis. The endocytosis only occurs when the virus binds to the receptor. And once it is in, inside the cell, the whole virus went into the cell. So it's not just the genome like with the bacteriophages, it's the capsid and the genome. So then the animal virus, the naked virus, has to go through a process of uncoding. And that is where the capsid is removed. So the protein capsid is removed, so then you end up with just the genome inside the cell. Now I'm just talking about a naked virus or a non-enveloped virus, and so I went through attachment and penetration. And if all you know about the virus is that it is a naked virus, that gives you no information on biosynthesis or maturation. You need to know what kind of genome is present for that information. But it does give you information on release. And naked viruses release by lysis. So they release by lysis, so the cell will rupture, and of course that will kill the cell. Cell cannot survive that process. Now I want to go through the life cycle of an enveloped virus. And so remember, knowing the outermost covering will give you information on attachment, penetration, and release. And as a reminder, the envelope is composed mainly of a phospholipid bilayer. So it's composed of a phospholipid bilayer, which is very similar to the cell membrane of the host cell. The other thing to remember is that embedded in that phospholipid bilayer are viral spikes. So these are protein spikes that were encoded by genes on the virus. So these are proteins, and they are considered spikes. So when the cell is going to attach, when an envelope cell is going to attach to a host cell, it will use these spikes to bind to receptors on the host cell. So this is an example of a virus, and this is the host cell. And here's the enveloped virus. So the envelope is on the outside here with those viral spikes. Inside is the capsid, and inside the capsid is the nucleic acid. So these viral spikes are going to recognize receptors on the appropriate host cell. So again, those spikes are used for attachment. Then penetration is going to be done by fusion. And fusion means that the envelope and the host cell membrane are going to fuse together. And that is fusion. And that is something that phospholipid bilayers can do. When you bring two phospholipid bilayers in close contact, extremely close contact, they will fuse together. So they will fuse together, creating one phospholipid bilayer like that. And when that happens, then the capsid with the nucleic acid can enter into the cell. And again, because the capsid has entered into the cell, this virus has to go through uncoding, where that capsid is removed, and then just the nucleic acid is released into the cell. So an envelope virus will use its spikes for attachment, then it will penetrate by fusion. Biosynthesis and maturation are determined by the type of genome. So if all you know about the virus is that it's enveloped, you don't know where biosynthesis and maturation are taking place. But you do know that this envelope virus will release by budding. So at the end of maturation, you have the capsid that has been assembled around the nucleic acid. And then that capsid will be transported to the edge of the membrane of the host cell, and then that membrane will start to bud out. And the membrane will bud out enough that you will pinch off a vesicle, and that is what gives rise to the envelope. So the envelope actually is derived from the host cell's membrane, and that is the process of budding. Knowing if a virus is naked or enveloped will give you information on attachment, penetration, and release. To know where biosynthesis and maturation take place, you need to know what type of genome the virus has. So now what I want to do is go through the DNA viruses. And on your list of microbes to memorize, you have several DNA viruses that you need to know. First are the herpes virus. So you have herpes simplex 1, which is the cold sore. Herpes simplex 2 is the STD, and also chickenpox. So remember that chickenpox is actually a type of herpes virus. Then another type of DNA virus that is on your list is hepatinovirus. Hepatinovirus, which I've mentioned before, can cause hepatitis. It affects the liver. Another one is papovavirus. Papovavirus causes warts, and those could be the warts that you see on the hands and feet. And also it includes HPV, human papillomavirus, which causes genital warts. And the last type of DNA virus that you should know are the pox viruses. And an example of this is smallpox. And this is an old picture of a child with smallpox because smallpox has actually been eliminated. So there are no natural cases of smallpox anymore in the world because we eliminated the disease by a process of vaccination. So these are examples of DNA viruses. Knowing that a virus is a DNA virus will give you information on biosynthesis and maturation. So if the only information you have is that the genome is DNA and you don't know if it's a naked or enveloped virus, then you won't have any information about how it attaches, penetrates, or releases. But you will have information on where biosynthesis takes place and where maturation takes place. So for biosynthesis, remember that there are two things that have to be produced. First is that the virus needs to make copies of its genome. So in this case, the genome is DNA. And in a normal eukaryotic cell, DNA is replicated in the nucleus. So that means that copies of the DNA virus's genome will be made in the nucleus because that's where all the enzymes are located. The second thing that has to be synthesized during this stage are the Pap Smears. And the Pap Smears, remember, are made of protein, so those will be made by the ribosomes. And the ribosomes are found in the cytoplasm of a eukaryotic cell. And remember, the cytoplasm is the area between the cell membrane and the nucleus. So these ribosomes are found on the rough ER and also floating free in the cytoplasm. So during biosynthesis, the genome, copies of the genome will be made in the nucleus and the Pap Smears will be made in the cytoplasm. So for maturation, you have to get these two components together. And if you remember when I talked about the nucleus, that DNA never naturally leaves the nucleus, but proteins can be transported into the nucleus. So for maturation to take place in a eukaryotic cell, that will have to occur in the nucleus, because that's the normal direction of transport. Proteins can move into the nucleus, DNA cannot move out. Papovavirus is the group of viruses that includes HPV and causes all the different types of warts, is a naked, so it just has an outer covering of a capsid. It's a naked virus with a DNA genome. And so now that you know the outer covering, it's a naked virus, and you know the genome, DNA virus, now you can describe the complete viral life cycle of papovavirus. So going through the first stage, for attachment, the papovavirus would use those protein fibers to recognize and adhere to the appropriate receptors on the host cell. Then it would penetrate by receptor-mediated endocytosis. After it penetrates, it will go through uncoating because remember the whole capsid penetrates. Then the genome, the viral genome, will be transported to the nucleus. And in the nucleus, that is where you will get copies of the viral genome made. And then the Pap Smears will be made in the cytoplasm. So biosynthesis is occurring in two locations. The genome is being replicated in the nucleus. The Pap Smears are made in the cytoplasm. Then for maturation, the Pap Smears will be transported to the nucleus and maturation will occur in the nucleus. Once the virus particles are all assembled, then the virus will release by lysis. RNA viruses are the other type of virus that I want to talk about. And with RNA viruses on your microbes to memorize sheet, there are lots of different types of RNA viruses, and I didn't have any good pictures of those. But for these, you have the coronavirus, which includes, of course, COVID-19. Filoviruses include Ebola. Flaviviruses include several diseases, such as yellow fever and dengue fever and Zika. Coronaviruses are known mainly for the common cold. Paramyxoviruses, measles, mumps. Orthomyxoviruses, influenza. Rhabdoviruses, rabies. And the last group, the retroviruses, they are technically an RNA virus. Their genome is composed of RNA, but they are a special group. So I will talk about them separately after I go through the RNA viruses. If all you know about a virus is that it is an RNA virus, then you have no information on how it attaches, penetrates, or releases from the host cell. But that does give you information on biosynthesis and maturation. So remember, for biosynthesis, you are synthesizing two things: copies of the genome and Pap smears. And for the genome is composed of RNA. And in the cell, the copies of the RNA will be made in the cytoplasm, so not in the nucleus. Pap smears, I talked about before. Pap smears are proteins, so those will be made by the ribosomes, which are out in the cytoplasm. So for maturation, both of your components are in the cytoplasm. The copies of the genome are in the cytoplasm, the Pap smears are in the cytoplasm, so maturation will also occur in the cytoplasm. The coronavirus causes many diseases like the common cold, and all picornaviruses are naked RNA viruses. So now that you know a picornavirus is a naked RNA virus, then you are able to give me the full life cycle. So naked virus will use its protein fibers to attach to the host cell, recognize and attach to the host cell. Then it will penetrate by receptor-mediated endocytosis. And also it has to go through uncoating because, again, the whole capsid has entered the cell. Once it goes through uncoating, then it goes through biosynthesis. Copies of the genome will be made in the cytoplasm. Pap smears will be made in the cytoplasm. So because these two components are both in the cytoplasm, you get maturation in the cytoplasm. And then a naked virus releases by lysis. Within the group of RNA viruses, there is actually a lot of variation in their genomes. Some RNA viruses, their genome can be made of positive sense. Some can be made of negative sense RNA. Some can be single-stranded. Some can be double-stranded. And all of those differences in the genome can have little effects on the viral life cycle. But that is a level of detail that I don't think you need to know at this point. But I just want to point out that no matter which type of genome the RNA virus has, always, with all of them, the biosynthesis is in the cytoplasm. So that doesn't change that general rule that I've given you. So RNA viruses always have biosynthesis in the cytoplasm and maturation in the cytoplasm. Technically, retroviruses are a type of RNA virus because their genome is composed of RNA, but they have a slightly different life cycle that I want to go through. There are many types of retroviruses, but the most famous one is human immunodeficiency virus, or otherwise known as HIV. And all retroviruses have the same basic structure. So all of the retroviruses are enveloped. So they do have that phospholipid bilayer, and they do have those spikes embedded in the phospholipid bilayer. And then, of course, like all viruses, they have a capsid, which is composed of the Pap smears, those protein Pap smears, And then, as I mentioned before, their genome is composed of RNA. Now, an additional thing that retroviruses have is they actually contain an enzyme. And the enzyme is called reverse transcriptase. And you know that's an enzyme because it ends in ase. And the name is actually very descriptive because this enzyme does the reverse of transcription. So if you remember from your basic genetics, transcription is the process by which DNA is copied into RNA. So what would the reverse of that be? It would be taking RNA and making a copy in DNA. And when that is done by the reverse transcriptase, we call that vDNA, little v DNA. That means viral DNA. So reverse transcriptase is an enzyme that will convert the viral RNA into a DNA copy. And animal cells never do that. So human cells never take RNA and make a DNA copy. So that is why the virus has to carry the enzyme with it, because the host cell will not have that enzyme. There is a second type of retrovirus on your microbes to memorize list that I want you to know, and it is murine mammary tumor virus, otherwise known as MMTV. And murine mammary tumor virus, this is actually the animal model for breast cancer. So that mammary tumor, breast cancer. And murine tells you what kind of animal it is specific for, and murine refers to mice. So like canine is for dog, feline is for cats, murine refers to mice. So if we want to use the mouse as a model for breast cancer, we need to infect the mouse with this virus, the MMTV, and then the mouse develops breast cancer. Now, a retrovirus is an enveloped RNA virus. So this will follow the life cycle, the basic life cycle for enveloped RNA viruses, but there will be a little twist to it. But in general, enveloped RNA viruses will use the spikes for attachment, for identification and attachment of an appropriate host cell. Then for penetration, they will penetrate by fusion. And then for biosynthesis, since it's an RNA virus, that will occur in the cytoplasm. And maturation will also occur in the cytoplasm. And they will release by budding. And budding, by the way, does not kill the cell because the cell membrane remains intact. So budding will not kill the cell can still remain alive. So a retrovirus will follow this basic life cycle, but the difference occurs during or right after penetration for a retrovirus. So as soon as the virus goes through uncoating, the reverse transcriptase is going to copy the RNA genome, the viral RNA, into DNA. So that's vDNA. Oops. So it's going to make a copy of the viral genome into DNA. And then that viral DNA is going to transport to the nucleus and it's going to splice into the cell's chromosome. So the host cell's DNA. And when it does this, it forms what we call a provirus. And when it does this, the host cell doesn't know anything has happened and the host cell can continue doing what it does. So it can continue metabolizing, moving, reacting. So it continues doing everything it normally would until there's some sort of trigger. And the trigger, we don't know exactly what it is with HIV, but it could be an illness, it could be aging, but whatever that trigger is, that is going to trigger the provirus to become transcriptionally active. So the provirus will not splice out, but it will become genetically active and its genes will start expressing proteins and producing the Pap Smears. You'll start making copies of the genome, and so basically it starts into biosynthesis at that point. And once you've started biosynthesis, which will occur in the cytoplasm, then you go to maturation, and then virus particles will bud off. This figure is an example of life cycle of a retrovirus. And again, the virus is an enveloped RNA virus. So it will use the spikes to recognize and adhere to the appropriate host cell. It will penetrate by fusion, then it will go through uncoding. And once it goes through uncoding, then you have the viral RNA will be reverse transcribed into viral DNA. So that reverse transcriptase will form the viral DNA. That viral DNA will get transported to the nucleus where it will fuse into the host chromosome, and that forms a provirus. So that provirus will sit there quietly for maybe the life of the cell, but if there is a trigger, and again we're not sure what that trigger is, then this provirus will become transcriptionally active. It will start being transcribed and the genes are expressed, and the cell will start producing the Pap Smears and copies of the genome in the cytoplasm. So you get biosynthesis in the cytoplasm, maturation in the cytoplasm, and then release by budding. One of the easiest ways to learn all the different life cycles of the animal viruses is to create a table. So in the first column, you can write the different stages of the viral life cycle: attachment, penetration, biosynthesis, maturation, and release. So these would be the first column. And then you can create or add all the different types of viruses. So the first one would be the outer covering, the naked virus. So the naked virus versus the enveloped virus. Naked viruses, remember, they use the protein fibers to recognize and attach to the appropriate host cell. They penetrate by receptor-mediated endocytosis. And for biosynthesis, you don't have any information about that. If all you know is it's naked, maturation, you have no information, and release, they release by lysis. The envelope virus uses the spikes. They penetrate by fusion. You have no information about biosynthesis or maturation, and they release by budding. Then you have the DNA viruses. DNA viruses, it gives you no information about attachment or penetration, but biosynthesis occurs in two places. The genome is replicated in the nucleus, and the Pap Smears in the cytoplasm. Maturation will occur in the nucleus because protein can be transported into the nucleus. DNA is not transported out. And release, you have no information. Then RNA viruses. RNA viruses, again, no information on attachment or penetration, but all the biosynthesis occurs in the cytoplasm, maturation occurs in the cytoplasm, and no information about release. So again, if you know the outer covering and if you know the type of genome, you should be able to give the full life cycle of a particular virus. And the last one I talked about were the retroviruses. And remember, the retroviruses, they are enveloped RNA viruses. So since they are enveloped, they use the spikes for attachment. They penetrate by fusion. Biosynthesis occurs in the cytoplasm, maturation in the cytoplasm, and they release by budding. But also remember, after penetration, you get that reverse transcriptase that makes the viral DNA, which splices into the chromosome and forms a provirus, which will sit in the chromosome inactive until you get a trigger. Once you get that trigger, then that provirus becomes transcriptionally active. It's genetically activated, and that will lead to biosynthesis, which leads to maturation and then release. When talking about viral diseases, we often relate to them in three terms that describe the type of viral infections they produce. And these three terms are acute, chronic, and latent. And these are referring to two things: how much virus is produced during the infection and how long the infection lasts. So the first term, acute, refers to a very short time. So this could be hours to weeks. And during that short time, you get a very high level of virus produced. So you'll get a very high level of virus produced over a short time. And some examples of this are influenza, the flu, and rhinovirus, the common cold. Also, another acute infection would be Ebola. That would also be another one. A chronic virus is one that lasts for a long time. So that would be months to years. So a very long time. And during that time, you get sort of a constant low level production of virus. So the individual is continually producing virus particles. And that would be several types of hepatitis and human papillomavirus. The last type is latent. And a latent infection can last a long time, but the virus production only occurs during short outbreaks. So for a period of time, there's absolutely no virus being produced. And that is a latent infection. All the herpes viruses do this. So the herpes simplex 1, herpes simplex 2, and chickenpox do that. Because viruses are too small to be seen with the light microscope, one of the ways we can identify a viral infection is by cytoplasmic effects. So these are changes we can see in the host cell. So for instance, this is what these normal host cells look like. And when you see a cell that has a different morphology, that indicates a viral infection. One type of cytoplasmic effect are inclusion bodies. And this is where you have cells that have darker staining areas in them. And that can indicate a viral infection. Those could be areas where biosynthesis is taking place or maturation. So these are cells. This is a normal cell, and this is an infected cell that has all these inclusion bodies in it. Also, there is giant cell formation. Giant cell formation is when many cells fuse together to form a giant cell. So if this is the normal tissue, often when these cells get infected with a virus, they will fuse together. And so what you will observe is something that looks like one giant cell with many nuclei in it. And that indicates a possible viral infection. A Pap smear is a well-known test which is used to determine if somebody may be developing cervical cancer. And what a Pap smear is doing is looking for cytoplasmic effects. This is an example of a normal Pap smear. So this is what a normal cervical cell would look like. And here they're just all kind of bunched together and folded on top of each other. But all of these cells look normal. An abnormal Pap smear would have cells that look like this. So that is a cytoplasmic effect where the cells have bunched up, they're rounded, and they no longer look like the normal cervical cells. And that's a cytoplasmic effect that indicates a viral infection, which could lead to cancer. Many viruses are associated with cancer. And this is a table which lists different types of viruses and relates them to the different types of cancer they are associated with. Probably the most famous one or most widely known one is HPV, human papillomavirus, which is a papovavirus. It's a DNA virus and it is associated mainly with cervical cancer. And that is why you get the HPV vaccine to hopefully prevent or reduce the chances of developing cervical cancer or any of these other cancers. But there are many other viruses that are also associated with many other types of cancer. And one of the things I want you to notice is that most of these viruses are DNA viruses or they are retroviruses. And that is very important because that relates to how they can trigger cancer. To explain exactly how viruses can lead to cancer, I'll actually start from the end result, from cancer. So I'll start at the bottom of the screen. And cancer, what is a basic definition of cancer? It's basically uncontrolled cell division. So what is happening is that those cells are dividing uncontrollably. So they have lost control of their cell cycle. So they're going through the cell cycle uncontrollably. And why did this happen? It happened because of a mutation. So at some point in time, there was a mutation in the genes that control the cell cycle, and that causes the cells to lose control and divide uncontrollably. And a basic definition of a mutation is a change in the DNA. So a change in the sequence of DNA, that's technically the definition of a mutation. And what can some viruses do is they can splice. They can splice DNA into the host chromosome. And when I talked about it before, I talked about the viral DNA from the retroviruses being able to splice into the host cell's chromosome. So retroviruses are capable of doing this. Also, not just the retroviruses, but also the DNA viruses. So if the DNA viral genome goes to the nucleus, it is possible for that DNA to splice into the host cell's chromosome, which will change the sequence of the DNA in the host cell, which could result in a mutation in the cell cycle genes and result in uncontrollable cell division. So that is associated with the retroviruses and the DNA viruses, because they do at some point have viral DNA which can mutate the DNA in the host chromosome, which can lead to cancer. RNA viruses don't usually do this because RNA cannot splice into the host chromosome, so it cannot mutate the DNA, so it's not associated with cancer. So that is why retroviruses and DNA viruses are often associated with stimulating cancer. The last group of microbes I want to talk about are the prions. And prions are basically infectious proteins. So they're not a type of cell, they're not even a type of virus, they're something new, and all they are proteins. Prions were identified first by Prusiner. Prusiner was doing his research in the 80s, and he actually received the Nobel Prize in the 90s for discovering prions. And prions are most famously known as the cause of mad cow disease. But these prions are just proteins, and so this indicates protein structure here. The type of disease that prions cause are called Spongiform Encephalopathies. So these are diseases of the brain and spinal cord, and it creates holes, giant holes in the brain and spinal cord, so it results in the death of neurons. So the type of side effects you see from these diseases is a loss of motor control, wasting away, and lots of neurological issues, changes in behavior. And all prion diseases are 100% fatal. They are also untreatable. So most individuals that are diagnosed with a prion disease have about a year before they die. Prions are relatively new microbes, so we don't know a lot about them, but the general hypothesis on how they damage neurons is depicted in this figure. So the first thing to know or to note is that a prion is a protein. And when you remember studying about proteins in a basic biology class, their shape is really important to their function. If you change their shape in any way, you will change their function. So the pathogenic prion, the disease type prion, has this particular shape. So it has one type of shape. And actually, all neurons have prion proteins, but they have a different shape. So all of our neurons actually express prion proteins on the surface, but this is the normal shape depicted by the box. So when a person is exposed to a pathogenic prion, so a prion protein that is folded in a different shape, that prion protein is going to interact with the normally shaped prion proteins. So it's going to interact with those normal prion proteins, and when it does so, it converts the normal prions into abnormal prions. So it's kind of like a domino effect. As long as you have one abnormal prion in your body, that's going to start converting the other prions into abnormal prions. And that continues, again, in a domino way or a cascade way. So when a neuron has all these prions on its surface, it's going to try to endocytose those prions to get rid of them. So the neuron is going to perform endocytosis, take them in, and it's going to create these vesicles with these prion proteins in them. And for whatever reason, it's very difficult for the neuron to break down these prion proteins. So what happens is the neuron starts filling up with these vesicles with the prions in them. And eventually the neuron becomes so full of these vesicles with indigestible prion proteins in them that eventually the neuron dies. And when the neurons start to die, then you get holes in the brain. Prion diseases were first identified in animals, and the very first animal that was identified with a disease were sheep. And the disease, when it's in the sheep, is called scrapie. And it's called scrapie because the behavior that you see in the sheep is they start rubbing against fences and trees until they start rubbing off their wool. And then even they can start rubbing to the point where they start rubbing off their skin. So again, that's very abnormal behavior, not normal behavior. And the sheep can become aggressive, and they also start to lose weight. And again, within a year of identifying scrapie in sheep, they will die. The second animal that has a prion disease identified are the cows, and this is maybe the most famous, mad cow disease. And the same sort of thing, the cows start to lose weight, they lose coordination, and they become very aggressive, which is unusual for cows. So that's the crazy cow. And again, once it's identified, they only survive for about a year. The last animal disease is chronic wasting disease. So chronic wasting disease in deer. And this is actually around northern Illinois, southern Wisconsin. And during hunting season, a lot of the deer are tested to see if they are carrying CWD. And again, you see a lot of wasting disease, loss in motor control, and so on. Prion diseases in humans. There are three basic types of prion disease in humans. And the first one is Creutzfeldt-Jakob. And Creutzfeldt-Jakob is a spontaneous disease. So when they say spontaneous, what they mean is they don't know what triggered it. So it seems to just spontaneously happen. And of course, the humans lose motor control, they have psychological changes, and eventually they go into basically a coma. And the only way you can really diagnose this is after death, when you do an autopsy and look at the brain and look at the spongiform aspect of the brain, so the damage that is done. But Creutzfeldt-Jakob is very unusual, but it's spontaneous. They don't know what triggers it. A second type of prion disease in humans is called Kuru, and Kuru is actually acquired. So you can acquire this prion disease by eating infected nervous tissue. And this was identified first in Papua New Guinea. In New Guinea, they had an epidemic of Kuru. And what they think happened in New Guinea is that one person probably developed the spontaneous Creutzfeldt-Jakob disease. And when that person died in these tribes in New Guinea, they had a funeral practice where all the family would gather and the friends would gather, and they would eat a piece of the person's brain. And that was their traditional funeral practice. So probably what happened is the one person developed the spontaneous form, Creutzfeldt-Jakob, that person died, the friends and family got together, they ingested part of the brain and infected themselves with the prion disease, and then those people died, and it just propagated that way. So this was a real epidemic of a prion disease. And once it was figured out how it was being transmitted through the population, the government outlawed that practice, that funeral practice, and then that stopped the epidemic. So Kuru is an acquired form of a prion disease. The last type of prion disease that I want to discuss is fatal familial insomnia. And the important term here is familial. So this is a disease that is found in families. And actually, this prion disease is a genetic disease. So individuals actually inherit a mutated gene that will produce the prion. And this gene is a dominant gene. So if you have a parent who displays this disease, you have a 50% chance of also inheriting the gene. And if you have the gene, you will develop the disease. In the world, there are about 30 families who have been associated with FFI. And the most famous family is a family in Italy. And it has been recorded for a couple hundred years that they have the symptoms of FFI. And one of the most recent members was a man named Silvano. And when he first started expressing the evidence of the disease, which you can kind of guess is insomnia, an inability to sleep, he asked the doctors to study him in hopes of finding a cure. But right now, there is absolutely no cure, no effective treatment for any prion disease. And like I mentioned before, they're 100% fatal.